started. It's my pleasure to introduce Talmo from the Salk Institute. Joining us today is going to be talking about quantifying and modeling behavior using deep learning. Talmo did his undergrad in University of Maryland, I think, which I did realize when I looked at the page today. And I first met him when he went to do his PhD at Princeton University, where he did a joint PhD with Joshua Shavitz in the physics department and Marlon Murphy in the neuroscience department. And I think, as you can probably see from some of these fantastic illustrations, it's this kind of combination of the biology with physics and computational approaches that makes Talmo's work so exciting to people. He actually did his PhD in the same lab as I did. I was the first PhD student in that lab. And one of the nice things about being the first PhD student is no one, for a brief period, you are unquestionably the best PhD student in that lab. But unfortunately, it didn't last long, and I was quickly eclipsed by Talmo, which really thought I had to leave the university. Just the shadow he cast was very long with his spectacular work, and it's no surprise that he was snapped up by the Salk to start his own group in 2021. So I'm sure he's going to tell us about lots of exciting work he's been doing since then. So over to Talmo.
Thank you. All right. Well, thanks for having me. It has been a delight meeting everyone so far. So today we're going to talk about behavioral quantification and some of the early work that we've done in modeling and capturing motion, to then how we think about motion generation and generative models of motion production. So let's go ahead and jump in. And to start, we're going to make a very sweeping observation about life, about living things, which is that arguably one of the main outputs of the evolutionary process has been movement, the function and the capability to move. Whether you're a plant going through soil, a fruit fly grooming its head, or a giraffe walking through a savanna, all forms of life evolve the ability to move at some time scale and length scale or another in order to meet evolutionary objectives to survive and escape threats, find resources, and pass on your genes. So if we want to understand life writ large, it would behoove us to have a good way to quantify these different kinds of biological motion. In other words, we want to be able to distill out these images and videos into a representation that might look a little bit like this, like capturing these essential degrees of freedom that correspond to the body, and the degrees of freedom afforded by the evolutionary constraints imposed onto the morphology of these animals. Okay, so today we're going to talk about some of the more technical work that we've done in what we call biological motion capture. In other words, how can we quantify these different kinds of movements that are produced by biology? And I'll talk a little bit more about what we mean by biological motion. And then in the second part, and we definitely won't have time to go through all of this, so we'll do a little pause halfway through, and we can choose our own adventure through a series of different applications of the core technology, as well as do you want to reserve some time at the end to talk about some of our models for embodied virtual animals, or our new approaches for modeling how biological motion is produced by nervous systems and the body. But to start, let's jump into this some of our earlier work on animal pose estimation and markerless motion capture. So first off, what do we mean by biological motion? Well, if we consider a movie that maybe looks a little bit like this, we have a bunch of dots moving around. Sometimes they stop moving. And whether it's a structured set of dots or even a single one, even when they're moving, even when we strip away all these visual elements, we can still say that these things are moving, but we wouldn't really think of this as biological motion. In contrast, let's consider an animation that looks a little bit like this, where initially it just looks like a bunch of dots, but all of a sudden, when these guys start to move in a particularly coherent and structured fashion, when you compare the visual stimuli that we're seeing on the right as compared to the ones on the left, even when they're both moving, very clearly, the one on the right evokes this perception of articulation, of coherence that follows a morphology. And essentially, we can perceive it. And we've done, and folks have done lots of psychophysical experiments where it's clear that you can report out what's happening under these animations. Fundamentally, they're all just a bunch of little dots moving around against a black background. We stripped away textures, we stripped away shape, but there's still something special about this kind of motion that allows us that there's something hardwired that allows us to perceive it as biological. So this essential representation is going to be what we're going to try to distill out. And later in the talk, we'll talk about why this representation is something that's so hardwired by interrogating the generative processes that give rise to these kinds of movements. We can measure biological motion at a variety of different levels of granularity, starting at the coarsest level, doing something like centroid tracking, center of mass tracking, that suffices to be able to pull out the dynamics that underlie certain classes of behaviors, such as navigation, where you are in space. But for other classes of behaviors, like say this fly grooming its head, it's clear that the center of mass wouldn't reflect enough of a signal to tell us about the kinds of degrees of freedom that are being actuated upon by the nervous system in this class of behaviors. So at this level, we need to be able to track not just where the animal is in space, but where all of its body parts are in space, where all degrees of freedom are. And this is what we call single animal pose estimation. And an even finer level of granularity, we need to be able to do this not only with a single animal, but with multiple interacting animals, right? This introduces a new set of challenges that fall under the umbrella of what we call multi-animal pose tracking. So what we'd like to do then is to be able to go from images and videos that look like this and extract out a set of landmarks that correspond to degrees of freedom afforded by the morphology of the animal's body. These points, these are what we call landmarks. They're going to be, for the purposes of this part of the talk, essentially all defined by the experimenter. And it doesn't really matter what these body parts are. We're going to here, I'm labeling them with some with actual textual descriptions, but in effect, we're just going to treat them as different classes of landmark types. So when I started working on this, there already existed, or there had just started to come out with some methods in the human pose estimation literature to do what's called markerless motion capture. And this is an example of one of these early systems that was able to do this using techniques from deep learning. And at the time, this was pretty radical of a paradigm shift given the complexity of the image processing challenges here. Sorry, let me just reshare. For example, here you have a dancer, you have a moving camera that's held, it's clearly handheld. You have movement in and out of plane. And would have given me nightmares my first year in graduate school when I was writing classical image processing algorithms. The subject is wearing black clothes against a black background, making it a nightmare to try to separate out those pixels from the background. So why did this work so well, right? The essential recipe that powered these systems and really served as the basis for all the work that we then developed was to start off with defining this big convolutional neural network. It takes as input an image of our subject, and it produces as output a spatial probabilistic representation of the location of each landmark. This representation turned out to be one of the key innovations that allowed these things to work really well. Essentially, what they are is a set of heat maps that here are overlaid in the original image. They're encoding the location of each body part within each one of these heat maps that each corresponds to a different body part that we can then use to extract the location of each individual landmark by finding the brightest pixel, a pixel that has the greatest probability of containing that landmark. Simple enough. The challenge was that while for humans there existed these massive data sets with hundreds of thousands of annotated images, anytime we had a new animal, a new body morphology, even a new camera with different image statistics in the lab, we'd have exactly 0 labeled images. And this induces what we call the label data problem, or reflecting a property of machine learning systems called sample efficiency. And here the challenge is, how can your system do well with relatively little annotated data, especially when it takes a long time to go in and click on all these points. So to address this, we first developed a system called LEAP. LEAP stands for Leap Estimates Animal Pose. It's a cute recursive acronym, courtesy of Diego Alderondo, my then undergraduate mentee. And we solved this problem of sample efficiency through network architecture design. So LEAP worked by taking a raw image, feeding it into a convolutional neural network that then learns to predict a set of these part confidence maps, just like the human pose estimation systems did. The key difference was that we hypothesized that if we design a neural network architecture that has the same kinds of inputs and the same kinds of outputs, but produces those using many fewer computations, many fewer layers that have fewer parameters, then ideally, the system should also require many fewer annotated frames in order to work as well as the larger scale systems did. And when we put this to the test, it indeed turned out to be the case. Even with as few as 10 labeled images, this thing started to do reasonably all right. For some body parts, it was doing quite well, and on others, it not quite as well. But even by the time that we got to around 250 labeled images, we were getting to pretty much as accurate as we would do, as any human would do relative to another. A byproduct of designing a network architecture that was lightweight was that it was very fast to train, which allowed it to then also predict on new frames very, very quickly, which essentially enabled this human-in-the-loop training workflow, where effectively we would train these models on a small amount of data, let's say 10 frames, predict it on tons of data that's unlabeled, correct the mistakes, and then train again. Going through this process, because every time you would go through the loop, it would require less and less data to, it would make fewer and fewer mistakes. We could generate more labeled data with even less time, right? Because we're essentially correcting fewer and fewer mistakes every time we go through this iteration. So not only does it require less data to begin with, it was also very fast to generate new data as you went along in the process. One of the downsides of having a lighter weight neural network was that potentially it has less capacity to handle different kinds of morphology, different kinds of image statistics. But pretty much everything we threw at it was working. So we kind of stumbled upon this recipe that seemed to solve these core technical challenges to being able to do this form of markerless motion capture. So then where do we go next? Well, we want to do this with, we started off doing this because we wanted to actually track social behaviors. In social behaviors, we need to track multiple interacting animals, which introduces a new set of unique challenges. While it was trivial enough to extend this system to detect not just one nose and one tail, to instead detect multiple noses and multiple tails, it was still very non-trivial to then figure out, okay, well, which nose should go with which tail? And we can't just pick the closest one, especially in the case of social behavior, when what we explicitly care about is when they're doing these close interactions. So we need to be able to model the relationship between those body parts. And this induces this first problem that we call part grouping. The second problem is what we call identity assignment, which is not just which body parts should go together, but which constellations of body parts should go together across times or across consecutive frames. And this introduces a whole other set of challenges, because now what we're trying to model is not just the location of a body part or what a nose looks like, but really what makes an individual unique and consistent across time, despite the fact that everything about their morphology is changing. So it's around the time that then we developed the successor of LEAP called Social Leap, or SLEEP. And SLEEP was intended to be an end-to-end framework to address these challenges using a variety of flexible approaches. SLEEP is a pipeline that takes you from raw videos through annotation, configuring train neural networks, to then train these models and, sorry, don't know what's going on with my laptop there, to then being able to track any number of body parts. Challenging. Sorry, I don't know what's going on with my video decoding on my laptop at the moment, but bear with me if some of these videos don't play. Okay. Here's a lot more videos that appear to be playing. Mostly. Unpleasant. Okay. Well, we try this out against a variety of different data sets, against a variety of different conditions. And like, once again, whatever we threw at it, a number of animals, a number of body parts, it all seems to work. But really, the key innovation that we put into sleep that has really contributed to its success has been paying a lot of attention and putting a lot of emphasis on the engineering of the system itself. Like I said, we need to solve these pretty challenging problems, part grouping, identity tracking. And at the end of the day, there wasn't a single solution that fit all. So in order to try out and create a scaffold upon which we could try out a bunch of different algorithms, a bunch of different approaches, we developed this as a very modular system, drawing from some of my experiences at Google AI in developing production-grade machine learning systems that could scale and that could be built upon. And as a result, we've seen a pretty solid adoption across different disciplines and different use cases. Now reaching a bit over 25,000 users, many of which are in this room, across pretty much all over the world, and an infinite amount of emails related to its usage. One of the big things that the folks really seem to like is this piece of the system here. And I won't have time to talk about all the different pieces, but happy to chat offline. One of the pieces here is how we integrate and annotate the data. And a lot of this is powered by this desktop interface that makes it really easy to just point and click, import your data, define the body parts you want to track, define a set of frames that you're going to annotate. It kind of helps guide you through which ones to annotate, to then basically be able to point and click, drag and drop, and determine the locations of all these body parts with lots of different hidden little shortcuts and user experience enhancements, largely driven by user feedback about what's going to save time, what's confusing about these options, so that you're not having to constantly switch back and forth to other tutorials, other documentation. We are also users of scientific tooling and deeply appreciate that no one reads the documentation. So we try to build in, anytime there is a question, we try to build something into the interface, into the design of the tool itself, guardrails, to hints, to whatever else, so that people just wouldn't have the question in the first place. And that has, I think, served us well, both as users and as maintainers of the tools. In any case, all right. So that's the general system. As I mentioned, I'm not going to go through all of this. I'm just going to highlight one quick capability here, which is around the performance. So one of the things we did was to make sure this thing runs really smoothly and really fast. So there's two different ways that we can improve performance along the speed axis. One is throughput. It's always great to be able to track more data faster. But at the end of the day, right, if your thing you can run the software over the weekend, and it can track all your data, who cares if it's 2 times, 5 times as slow? But where speed really starts to make a difference is when it comes to real time. Where here, there is no shortcut to it. You're trying to minimize latency. What is the time that it takes between when you first get an image to then be able to process it? And through a variety of innovations, we were able to basically run this without any optimization for real time, just by using a batch size of 1. That is, we're processing 1 image at a time. We're able to get to around 12 milliseconds of latency on a 1024 by 1024 pixel image. It's a high resolution image, something that's necessary to balance having enough resolution on the individual limbs that we care about, and having a large enough field of view that it doesn't affect the behavior by having too constrained of an arena. And we get to pretty good latencies. But we worked with some folks at NVIDIA to adapt our models to be able to be compiled to an inference engine called TensorRT, which is optimized for inference on NVIDIA GPUs. And for the throughput, This essentially tripled the performance with effectively no change to the models or sacrificing any accuracy. But for the real time, it really blew us away that we could even achieve these kinds of latencies. But we were able to get down to around 3.5 milliseconds. This is even on a 1080 GTX Ti back in the day. And there's no special tricks here. I mean, there's a whole bag of tricks here that happen under the hood, but there's nothing that special that we do to optimize for real time as compared to our offline or throughput maximized processing mode. And as a proof of principle that we could leverage this in a real experimental system, we use it to detect a courtship approach of a male fruit fly paired here with a virgin female that is predisposed to accepting courtship advances. Now, because we can detect based on the pose that the male is approaching the female, we can optogenetically activate a set of neurons in the female's brain that essentially controls her ovipositor extrusion. This is her mechanical rejection response. But in other words, we're able to detect when a male is performing a courtship approach, and we're able to artificially control the behavioral response of his social partner, right? Demonstrating that we can do not only behavioral closed loop, but behavioral closed loop across animals, allowing us to basically patch into this social behavior response. And to make this even more accessible, we worked with some folks on building a bonsai integration for this so that you can run it as part of this visual programming language bonsai that then allows you to couple the real-time tracking with experimental control. Sweet. Okay, so where do we go from there? We have a couple different axes that we're pushing along. One is to improve our ability to do this identity tracking. I didn't talk too much about the identity tracking that we have in sleep right now, but while it's very good, it still always has room for improvement. Identity tracking is one in which you really can't have any mistakes, so any amount of improvement always helps. And one of the things that we began to appreciate was that we can kind of recast this problem as a pure multi-object tracking problem that allows us to create an architecture that allows us to leverage multiple different kinds of cues in order to do identity tracking. So we came up with this transformer-based architecture. Transformers are a special kind of neural network architectures that essentially can better reason about sequential data. And so what it's able to do is to take in a sequence of detections. And here, we're going to abstract away what detections are. It can be a single point. It can be a bounding box. It can be a whole pose, or it can be a segmentation mask. Dream stands for dream relates every entity's motion. Dream doesn't care about the nature of the modality of the detections. All it's going to do is it's going to integrate where the detection is in space, in time, and the visual features, and stitch all that together to output a set of associations. Essentially, associations are how related are different sets of detections. This is essentially the core thing that we need in order to be able to associate detections across frames by learning to mediate between these different cues. This really helps in complex situations where we have lots of entities and complex imaging conditions. And the accuracy metrics are great. It's a drastic improvement over sleep and other methods. But essentially, to demonstrate the generality of this approach, we've been applying it to lots of different modalities of data, ranging from multiple animals in, say, like a home cage, to a bunch of fish in a dish, to even dividing cells at different levels of density. And I don't think I have a video here, but going all the way down to tracking individual organelles within neurons in live microscopy. In essence, it all boils down to the same problem of relating detections to each other. Well, we've done really good in detections. This axis of connecting those detections to each other has been still an open challenge. So kind of excited that this approach is going to give us a significant boost, especially because these performance metrics I was just showing you all are largely a zero shot, meaning you don't need to train on any new data. We have some pretty strong generalizable models that are going to work across modalities. Okay, great. So that pushes us on the time axis. What about space? Well, one of the first things we started working on when I started the lab was to start to extend this from 2D to 3D. And the first pass initial approach that we took essentially just takes multiple views of the animals. We do the 2D pose tracking. We have calibrated cameras. And we use that to relate the geometric information across cameras to each other. We have to solve a couple of different challenges here. One is that some views are a lot harder to track than others. These side views in particular can be a bit challenging. There's a lot of variability in scale as compared to the top views. And then more fundamentally, we need to solve this cross-view association problem, which is way harder. Essentially, we need to be able to reason about which animal is the same animal across different camera views, especially when they look identical, and there is more of a change in their appearance just by having cameras placed at different angles than there are differences in appearance across the animals themselves. So that becomes a bit of a challenge, and we need to be able to take both the geometric information and appearance information into consideration. Our first pass naive approach here, we have a new thing that I won't really talk about today, but the first pass approach has been to basically leverage with some constraints an exhaustive but heuristically constrained search of all possible configurations of which animals should be corresponding with each other animals. We triangulate them in 3D, and then we compare to how consistent they are with the 2D projections. And there's going to be some challenges there and limitations, especially when it comes to scaling to larger numbers of cameras, large numbers of animals. But this, with some optimizations, actually works pretty well. We have a new approach that is able to reason about this and leverage time to further constrain the problem. As part of this, we've developed a pretty large-scale data set of around 2 million synchronized frames across 8 cameras that we're pretty excited will serve as a pretty solid foundation for continuing to develop these kinds of methods. As a proof of principle here, we have one short clip from one of our example sessions that shows us reconstructing the relationship between these different animals in a standard kind of a wedge enclosure here. And what's nice about analyzing social behaviors in 3D is that by putting all these key points in metric space, we're able to reason about the likelihood that they're able to, say, perceive other socially relevant stimuli, such as odorants that have a reasonably approximatable length distance of how far away they can perceive volatile odorants. And that allows us to essentially then detect and pull out all these dyadic social investigation events, and then align that to their kinematic signature. And one of the things that we find is this interesting dip of their snout when we trigger on these investigation events. And interestingly, it happens both when they're doing anogenital sniffing and orofacial sniffing. The orofacial sniffing doesn't necessarily need to involve a dip, but we think that it might be related to an underlying motor program, and we're continuing to investigate this. But the point being that we're able to now have really high resolution and acuity on the motor signals that underlie the social investigation, just by being able to triangulate these guys and reconstruct these complex naturalistic interactions in 3 dimensions. Okay. Good. Take a very quick pause there. Okay, so we have a few different directions to go in here. I'm going to make a call here based on my interactions thus far. I think I want to jump to our embodied animals before we end with maybe a couple of different vignettes on some of the more eclectic applications of these systems and tools. So let me skip ahead here. Virtual animals. Okay, so first off, just want to acknowledge that this has been a very long journey over the past 3 years or so, where we've been trying to come up with different models of biological motion production, and it has very much taken a village. A lot of the work that I'm going to be showing you guys now is largely unpublished. We're just about to submit the first pass of this, but a lot of this once again to Diego Alderondo, who after Princeton went to do his PhD with Benzo Ovechsky at Harvard. Charles Zhang, who has been leading the efforts on the Harvard side. Scott Yang in my lab. Eric and Elliot Abe in Bing Brunton's lab. So to start off, I'm gonna make this provocative statement that despite my lab specializing in developing tools for key point tracking, I will be the first to admit that the brain does not control key points. If we want to understand behavior and how the brain produces it, we need to acknowledge that the output of the brain does not directly feed into X and Y and sometimes Z coordinates of all your body parts. So what is the process for biological motion production? Normally we have a conceptual picture that looks a little bit like this, where we have a central nervous system that sends out motor commands, and some magic happens, and then we have behavior, right? We have all these points in a plot. Sometimes we might start to introduce a little bit more about this side of the picture. We consider in our models different sensory modalities, how the time course of different sensory stimuli can then influence and shape through a cascade of processing in the central nervous system that then will influence what motor command you're going to output next. What we often ignore is something that is highly motivated by this video. So this is not tracked, but doesn't matter, right? What do we see when we look at this? Right? We see pretty complex movement. We see undulatory, oscillatory, periodic movements. It might be suggestive of all sorts of neural control of movement strategies, central pattern generators, closed loop interactions with the environment, right? We can form all sorts of neural hypotheses about how a fish is able to produce these kinds of motions. There's even a strong ethological basis for why they'd be able to move and undulate in these particular patterns. And the longer we look at it, the more patterns we're going to find, right? Different kinds of swimming modes, different kinds of turning, and so forth. Well, the kicker here is, of course, that this fish is dead, right? There is zero nervous system activity happening here because the researchers here went to the market and literally got a dead fish from the supermarket, dropped it into this tank, and then imaged it. So what this is supposed to invoke is the fact that not all of movement production is produced by simply through motor commands, that everything is just driven by the brain. In fact, quite a lot of intelligence is what we call mechanical intelligence. That there's a lot of movement patterning and structure in behavior and biological motion that is shaped by the priors and dynamics that are produced by the interaction between the body and a physical environment. And ignoring all the processing that happens downstream of the nervous system is bound to be missing a massive part of the picture here. So having now developed these tools that allow us to capture biological motion with the ultimate goal of understanding the brain, we now need to acknowledge that there isn't a direct transfer function. We need to acknowledge what happens in the middle, which is the musculoskeletal system and the external environment. So how do we go about modeling those two? That should be easy, right? Get all the key points already. So how hard could it be? So in this part of the talk, I'm going to focus a lot on how we've been working towards modeling this end of the picture and leveraging that to better understand how the brain is able to produce biological motion. And so to start, we needed to come up with models of the body. And around the time that I was starting my lab was when some of this work has just started to come out from Benzo Ovechsky's lab and in collaboration with Google DeepMind to develop these musculoskeletal models, well, more skeletal models than muscular, of a rat that is compatible with running in a physics engine called MuJoCo. MuJoCo is normally used in robotics, and importantly, what they were attempting to do was to model how the brain produces movement as guided by a biomechanically realistic model of motion production. And so the way this is going to work is to have a set of artificial neural networks that take in sensory inputs of perceptions of the environment and produce a set of motor commands in the form of actual joint torques that get applied to this actual articulated biomechanical model. And what these guys were able to do was to demonstrate that if you take an artificial neural network, drop it, allow it to control a realistic, semi-realistic rat body, and then ask it to produce, behaviors that, you just drop it into different tasks that rats can also do, such as navigating out of hilly terrain, running down a corridor, not falling into these gaps, foraging across a maze, and so forth, that they were able to, model how much having a semi-realistic body can induce different kinds of behaviors that we also know real rodents can do. So this was a significant step towards demonstrating that we can begin to model biological mechanical intelligence in a realistic fashion by demonstrating that you can use a realistic body, semi-realistic neural controllers in realistic physical environments. Okay, yeah. Yeah, so the skeleton, the masses, and an approximation of the degrees of freedom through torque-based actuators. And I'll talk for each limb. Yeah. And I'll talk a little bit more about how we're advancing the biomechanics there. But okay, we have these guys. The question is, okay, how can we then improve upon the models of how the brain is producing motor control? So to start off, when we initially reproduced this, we found that, okay, great, we can, put in a virtual camera in the head of the animal. We drop them into this environment. They have a realistic body. We're able to train a neural network that takes these sensory inputs in the form of vision, mechanosensation, proprioception, and it learns to produce a set of torques applied to the body to solve this task. Where here, one task is escape out of this hilly terrain. Another task, it is run down this corridor and don't fall. And they are 100% doing what we're asking them to do. They're getting maximum reward, maximum score against this behavior task, right? And if I wasn't looking at the actual videos of them doing the thing, I'd just be looking at the readout, I'd say, awesome, my rats are super smart. They're able to fully figure out how to do the task. Yeah.
Movements that try to conserve energy.
Yeah, so then we can start to play all these games, right? Of like, okay, well, I can give it this reward that just says like, you get more reward for leaving this hilly terrain, and maybe if I further constrain and say like you have to use your forelimbs or you are not a kangaroo, maybe don't touch the back of your body against the floor, and so forth, right? Maybe minimize the amount of energy that you're using. Eventually, you're going to get to approximating more and more realistic looking movements. The problem is you can, spend like multiple PhDs worth of work coming up and crafting a reward function that tries to optimize against every possible constraint that these animals do that gave rise to the kinds of movements that we actually see in nature. The problem is those were really shaped by their entire evolutionary trajectory, right? There are many constraints that happen across their evolutionary trajectory that led them to move in the way that they eventually learned to move. Some of it is baked into the body, some of it is baked into the brain, and some of it's learned just even during their lifetime. It is entirely possible for us to craft these reward functions, but at that point, I'm not really sure what we're really learning in terms of the biology. And indeed, if our ultimate goal is to model how the brain is producing complex behavior and complex movements, it would be great to be able to, at a minimum, be able to constrain it to what is actually observable of their behavior. Like sometimes they're energy optimal, but a lot of times they aren't, right? Evolution is not a perfect process. It is very often, pretty imperfect. It often, gets stuck in lots of local minima. And while minimizing energy consumption is certainly a reasonable general goal, it is not necessarily always the case that every animal and every biomechanical embodied system is optimal in every setting. Rather, we don't want our agents to be the perfect, idealized rodent. We want them to be a rodent that looks like the rodent, moves like the rodent, and computes like real rodents do. So in order to do that, what we're going to do is not make assumptions about what their objectives functions were, but rather we're going to say, well, we know what real rodents look like when they're moving because we can track them. So we're just going to say, move like real rodents do. So to do that, the first step is to do what's called inverse kinematics. And so this is a technique from computer graphics and often used in animation. Essentially, what we're doing is we're taking these key points and we're going to essentially try to optimize for the optimal pose configuration of the body model that is most consistent with the key points. They might not align one-to-one. There might be some offset. There's going to be some noise. But we've worked out an algorithm that is robust to lots of these cases. We're even able to give it artificially jittered and noisy key points, as is often the case. When, let's spend a lot of time optimizing your motion capture system. And it's still able to essentially clean all that up by leveraging the inverse kinematics to ensure that the only plausible poses that are realistic with the morphology of the animal are actually produced here. OK, so we've now essentially translated our potentially noisy key points that are derived from real data to the body model space. What we're then able to do is to play that back. We're going to be demonstrating to our agent, to our virtual animal here, what the sequence of movements are that real animals produce. And we're going to ask it, we're going to give it the task, imitate what the real animals are doing. OK? They're essentially going to do what in some senses could be called observational motor learning. Right? We're going to show them what a real movement looks like. And much in the same way that look like a very sporty crowd, right? So imagine your football coach is showing you how to dribble, right? You can observe it, you can see it once. It doesn't mean that you can do it, right? You still got to practice and do that many times. So what we've done is we've come up with a network architecture system that is kind of compatible with this form of learning. Essentially, what it does is it takes a target trajectory that's derived from motion capture of real animals. We're going to encode that into a fairly low-dimensional Gaussian distributed latent space, essentially some vector that represents a sequence of movements. And we're going to then train a decoder neural network. And this decoder, essentially, think of it as analogous to the motor system. It's going to get shaped by some intention. It doesn't really care what shapes that intention, whether it's observation or whether it's sensory inputs from a given task. And it's going to learn to decode that out into a set of activations or actions that get translated into motor commands, something like joint torques or muscle activations, depending on the biomechanical model. It gets feedback from the environment because it learns to output a motor command. The motor command gets integrated through the physics simulation to update the position of its body in an actual physical environment, virtual physical environment. And then it gets a higher reward for realizing the same trajectory over many time steps that the real animals was producing. And as you can see here, like the little white ghost is the target that's derived from the real animal data, and the colored model here is the movements of our virtual agent. As you can see, you can track it pretty well. You can even do this under some settings that are fairly challenging for motor control, like these being able to rear and balance on just two hind limbs, switch between different movement modes, walk around, run, etc. Writing down a set of control equations that would allow you to be able to move like this would be remarkably hard, is in fact the missive of the entire field called robotics. And it is incredibly challenging. And really, a lot of the techniques that we're borrowing here come from robotics indeed, except that our ultimate goal is not to then download this policy, this neural controller, into an embodied physical model, but rather we want to use this as a lens into how real biological systems perform these kinds of movements. And what we see is that now when we first train this neural network to essentially be able to move like the real animal does, and then we drop it into this task, what we see is that rather than adopting a more creative motor control strategy, it's able to instead reuse the motor primitive that it learned by watching how real animals move. So not only is it able to solve the task much more quickly and efficiently, it's doing so in a way that looks a lot more naturalistic. Still not perfect, but certainly much closer. So now that we have a model that can essentially move like a real animal, control a realistic animal body, and can be controlled using artificial neural networks that can be structured to look like the brain, what we essentially have is what we're calling virtual neuroscience. This is different than computational neuroscience, which attempts to model specific, sub-processes and different parts of the brain to different levels of realism. Here, our goal is explicitly, we want to model exactly what's happening, in vivo, in real animals with direct analogs. Our entire goal is to create and put on equal footing models of the brain, the nervous system, of the body, of behavior, and of the physics and the environment in which it operates. And this was funded up until quite recently, when it fell victim to the whims of the US government. But we're still committed to the vision and mission of the Virtual Neural Lab, and we hope to continue to develop this out into a platform that everyone can use. But just to preview a little bit about what we can do with it, as I mentioned, we can create these different tasks, right? We can train these models to already learn how to move like real animals and then drop them into more complex tasks. And by having artificial neural networks that control them, we can interrogate everything about their underlying kind of a higher level, more cognitive strategies. We can see how they weigh different sensory streams to mediate and inform their actions. We can manipulate it. We can inject different perturbations, mechanical, neural, and behavioral, like at the task level. We can, yeah.
To understand what exactly controls the body, can you go back maybe
Yeah.
slide when you have the neural network?
Mm-hmm.
Yeah, here, so here it is when it doesn't make any sense, you can just keep it and you can talk about it. Here you're using this to train the simulated brain to produce natural behaviors.
Yeah.
What part exactly do you keep as the brain that you then train on the task?
Mm-hmm.
I missed that part.
Yeah, no, I fully glossed over it. We keep the decoder. Yeah, and the decoder just takes this Gaussian distributed high-dimensional random variable that can then get decoded out into a set of commands. What we feed into this layer can come from different sources. What sets up their intention can come from either observation of a target trajectory, but it can also come from different sensory modalities. So yeah, like I was showing earlier, we can have, say, like a visual encoder that takes in a rendering of their visual field, extracts image features from that, and use that to set up what the intention is. It can take in different sensory layers, right? Sensory streams. It can take proprioception, mechanosensation. Audition and olfaction are a little bit trickier given the, how to simulate that in this physics environment, but it has been done, and we're generally working towards that. But essentially, we can have different kinds of encoders that all get shaped together, get integrated to essentially output into this motor intention latent space.
Latent space after to look at the representations of movement.
That is exactly one thing that we can do, and I'll show you what that looks like. So here is another body model that we've been working on. This is to get into the more nitty-gritty of fine scale motor control. This is in mice rather than rats. We're able to do this inverse kinematics from multiple views, and then train them on these tasks to reach to a particular target, varying the size and location and distance of that target in space, and then look at that latent intention space, for example, to find the geometry of their representations of those trajectories, align them to different phases of the task, and compare how this higher level representation compares to, say, the structure of the geometry of the actual kinematics in comparison to the control signals. So this can be used by itself to find interesting representations of behavior unto itself. And in that case, you could even, let's say, leverage the encoder alone. We're upping the realism of the body model itself by including not just the skeleton, but also muscles. Muscles are a lot harder. They increase degrees of freedom by a significant amount. And it does introduce more complex control strategies. Is it a 3?
Sorry, you're in lecture theatre 2? Yes. Is it here?
It is here. We're almost done. Okay. Muscles are hard. So we're demonstrating that we can do this in rats, in flies, and in mice. So here's one that has a lot more degrees of freedom. This is a body model developed by Srini Turaga at Janelia, again in collaboration with DeepMind, and we've been enhancing it, adding muscles and all sorts of capabilities to it, together with Bring Brunson. And I'll also just mention that we can, by virtue of the fact that these are artificial neural networks and neural controllers, we can basically inject our own control signals to drive this interactively. So what we're doing here is we made a little what's called a joystick policy, where we're able to essentially take in these command inputs here on the top left. This is from a literal, like, PlayStation controller. We're able to read out from the PlayStation controller a forward rotational velocity that then we map onto that intention space. How that gets decoded out into movements is entirely trained on real data. So now what we've done is developed out our Grand Theft Drosophila, which is really our end game all along. Well, in the interest of time, I suppose, maybe I'll stop there and take a couple questions that maybe might prompt some of the bonus stuff. But otherwise, let me just very briefly flash my acknowledgement slide here before we jump into the many bonus slides or get kicked out of the room, I suppose. Nope. Do we have enough bonus slides? Okay, good. Thanks. Yep.
Can you give us some idea of the differences between the two for you're focusing more on the social aspects? How do those software
Sure, yeah. So the core approach that we take in sleep has been in the network architecture design, takes the sample efficiency problem, whereas Deep Lab could originally leveraged transfer learning as the core technique. The downside with that is that you're kind of constrained to the network that was pre-trained before. Lightning Pose kind of has an interesting balance between leveraging more modern techniques for improving sample efficiency by leveraging constraints like the statistical subspace of plausible poses and temporal information, which we do not in sleep. They, on the other hand, don't support multiple animals super natively. And beyond that, it's mostly about the accessibility and usability of the software. Like at some level or another, you can kind of tune all these guys to kind of give you an equivalent trained network. But I think it ends up being more about usability. I think these days, increasingly, I feel like pose tracking in particular is definitely a lot more commoditized. Like you're really, there's a variety of options. I think the thing that works the best and the quickest is probably the best solution. Oh, okay, fantastic. Great, yeah. Yeah, you go first.
Just one small question. How well do you think that sleep or dream will quantify low contrast animals like C. elegans and they are non-vertebrates. So
Well, for C. elegans in particular, there's a whole host of different approaches in the worm community that have been like really, I think, well tuned for these kinds of settings. I point you to the work of like Evyatar Iamini and colleagues. They look quite good. There was a good one in Nature Methods, I want to say last year, that dealt with some unique challenges that you find in worms. But in general, all these things will work pretty much just as well as each other. There's nothing that specific in sleep that I would say is specific to this. The low contrast problem is a just general SNR problem, right? And there, it's just about tuning your tracker to adapt to those settings. But the deep learning kind of like glosses most of that away. It doesn't tend to care too much about it as long as it's visible even to you. Beyond that, you just need enough data.
assigning the key points and the coordinates.
Yeah, so right. So then you're kind of alluding to the fact that it doesn't have like a rigid, let's say, skeleton, right? And towards that, what I'll mention is the stuff that I skipped earlier, which is some of our plant work, right? We found that we're able to do this using some creative annotation schemes in a variety of different kinds of data sets, of time lapses of plants in a variety of different settings. You do have to get a little creative about how you define the skeleton when you don't have like a rigid endo or exoskeleton, let's say. But it all works. And essentially, we're able to demonstrate in a paper last year that you're able to use the exact same approach. And here, what we do with these kinds of images, these are plant roots grown in agar, is that we're able to define each root as being its own instance of a skeleton. And that way, the skeleton just becomes a set of landmarks. In the worm, think of it as like the center line, essentially. And so it works. You can just come up with your own strategy. Either they're equidistant, or you kind of spread them out with fixed lengths. And it's still able to adapt. Sorry, anything over there? Yeah.
Nice talk. So, about your like virtual neuroscience stuff, your embodied thing. So you've gone to like a lot of effort to actually simulate the body, like the affordances of plants and movement, right? It's really interesting. But then the other part of the virtual neuroscience thing is that you're just using the convolutional deep network to sort of control that body. But convolutional deep networks aren't really like brains,
right?
Like very often. Right?
Nope.
And so what do you think we learn then in terms of like coming at it from the point of view of a neurophysiologist? What do you learn about what that network does? How does that tell us about the brain?
Yeah, that's a fantastic question. And so think of this as definitely a starting point and a scaffold. And really what this is our foray into this field of neuro AI, right? Which is this more modern take on using artificial neural networks to model biological neural computations. And there we have a few different axes along which to constrain these guys. And one of them is, let's say, the topology, right? So we can structure these neural networks to not just look like a generic black box, but rather to be wired up so that topologically they look like the connectivity that we see in the brain. We know a lot about this in certain species from, say, ultrastructure level connectomics, so synapse level connectomics. And what we're working towards now is essentially dropping into these neural networks topologies that essentially directly mimic what we have from the connectome, in particular in the fly and the worm. Then we have one-to-one mappings between units of this artificial neural network and that of the biological neural network. But unlike previous systems, like think of like this Yemen's, Dan Yemen's, Jim DiCarlo kind of work in modeling the ventral visual stream using artificial neural networks, the output is still biological, right? So then we can ask questions about how we can trace through this entire neural network, let's say how a sensory percept, like say a visual cue, gets transformed through every part of the brain and converted into an actual motor command output. So as a neurophysiologist, you're able to then say, okay, well, I can see what the neurodynamics are at this particular, circuit of interest. I can now ask, what does that get translated to? What are the plausible solutions for how that information gets transformed and computed 2 synapses down, 3 synapses down, and then translated into the motor command? And importantly, because it's an artificial neural network, you can stick an electrode wherever you want and record activity in the entire brain, the entire nervous system. You can perturb using complex stimulation patterns that would be impossible to do in a real living system. And you'll still get the behavioral readout no matter what, no matter how many electrodes you stick in there, right? No head fixing necessary. Yep.
So, I mean, I don't know how sensitive the videos are. Even in the more realistic kind, it was not using the scoring to it. And I was wondering whether there was something about the network architecture that was compromising that.
Yeah, that's a really good question. That is challenging because while when we ask it to like imitate what the real animals are doing, we see that it is indeed able to adapt to these different, very realistic looking gait patterns, right? The second that we drop it into this other task, it kind of reverts back to this galloping phase, right? Part of it is because we are just training them separately. But technically, like the motor controller itself, the motor controller that's able to do this is frozen. It does not change in the task. It just, in order to solve that task, just decides to do this, to use more of a gallop-like gait. This is where we could try to bring in the energetic penalties again. We could try to impose these other constraints, but it kind of goes, that's kind of beyond the point, besides the point, right? What we want to do is either through the network architecture, through baking in these motor skills, or through different kinds of learning paradigms or behavioral task objectives, try to induce the similar kinds of behaviors. But our working hypothesis is that, like, if you see a rat trying to run down the corridor as fast as possible, it's indeed not going to, very slowly and meticulously, have an alternating gait. It will try to gallop as fast as possible. So I think it's just something that's permitted by the biomechanics, and so it's able to do that. But between, strategy here on the left and on the right, we know which one is most likely to produce hypotheses about motor control that is more likely to translate to the biological system. You got it. Cool.
Good.
All right.
Thanks, Talmo, again.
Okay. Great.
Not sure if it's a question.